Welcome for this session. Today we are going to discuss about variation, lesson 2. I hope last time we had a session about variation, lesson 1, and I hope you remember what is meant by the term variation and some other key concepts within variation lesson 1. So today we are going to discuss about variation in lesson 2, so which means it is a continuation of the last session we had. So when you look at this graph, you will find that there is a big difference between continuous variation and discontinuous variation. When you check on y-axis, that's where we have the percentage of population. Then on x-axis we have ABO system, blood group system. We have blood group A, individuals with blood group A. Basically we have almost 90 people in the whole population. Again, you could find people with blood group B, they are basically 60. They are basically 60%, we have 60. Then those with blood group AB, when you check on the graph, you could find that the number goes down. We have 40. Then individuals with blood group O, basically they are also correlating with individual with blood group A. But still, members of blood group O, you could find that now the peak of the curve goes higher than the peak of individuals with blood group A. So, in brief, discontinuous variation, there is a clear-cut difference, unlike continuous variation where there is no clear-cut difference. Another major difference between continuous variation and discontinuous variation, you could find that in discontinuous variation, basically there is no intermediate forms. But for continuous variation, there are so many intermediate forms exist within the population. But in discontinuous variation, we don't have people who are falling in the middle of the range. Okay, so that's why even from this graph, you could find that there is a clear-cut difference between individuals within population. So let us now move to the causes of discontinuous variation. Number 1, chromosomal aberration. We have different forms of chromosomal aberration. There is deletion, we have duplication, we have inversion, and again we have translocation. Change in chromosomal number through aneuploid and proploid. So basically, starting with deletion, you understand that's where you could find genes being lost from the chromosome. There is also doubling of genes in the structure of a chromosome, that is duplication. Inversion, that's where the arrangement of genes within the structure of chromosomes could be now the reverse way of it. Then there's this condition where you could find the whole set of chromosome is multiplying inside the cell. So that is what we call proploid. So now aneuploid, you could find a half number of chromosomes, basically which does not reflect the normal number of chromosomes we know here. Again, more ways to that, there's also a change in gene structure and expression due to addition, deletion, or change in nucleotides. So these forms of chromosomal aberration, once we finish variation, we shall move to the next unit, which is mutation. I hope that's where you will understand basically what these different forms of chromosomal aberration. But variation and mutation, there is a link, there is a clear link between these two units. So when we start mutation, I will explain more about these differences, these forms of chromosome aberration. Crossing over during prophase 1 of meiosis. Orientation of chromatids during independent assortment of metaphase 1. You have even had that one in continuous variation. Random fertilization of gamete, random fusion of gamete, and the effect of genetic segregation and the genetic reassortment. So all these are the major causes of discontinuous variation within a population. Let us again move to characteristics of discontinuous variation. So number one, like in every population, how could you understand, how could you tell somebody that now within this population I could analyze different individual organisms that now are falling under discontinuous variation? One of the feature has distinct categories for organisms. So you have had in a discontinuous variation, there is a clear-cut difference, okay, within the population. So you could find there are so many distinct categories for organisms, for sure. Again, discontinuous variation, they are either present or absent in the individual's population. This is true. Look, remember we have talked about polydactyl, the extra fingers, the extra toes. So once you move from one population to another population, you could find, in some population, you could find those individuals who have such a kind of abnormalities, but others, they don't. So that's why they say most of the features of discontinuous variation, they are either present in a population or absent. Discontinuous variation are controlled by a single gene or allele. So that's why in some population you could not find people who are polydactyl, people who have extra fingers, extra toes, earlobes. So it is as a result of the cause. So that's why they say that discontinuous variation is controlled by a single gene or allele. They are not measurable. That's why, in other words, you can also call discontinuous variation qualitative variation. It is similar. Again, as you have heard from the beginning, in a discontinuous variation, there is no intermediate forms. There is no intermediate conditions. So what is basically there, you could find a clear-cut differences among individuals within the population. So that is discontinuous variation. Now, this is a nice, here we have a clear summary between continuous variation and discontinuous variation. It is a table which summarizes properties of each form of variation. So what you can do here, you can just take your time, then you go through each property, you link it with another property in terms of continuous and discontinuous variation. Again, it is from this table where now the whole summary about continuous variation and discontinuous variation is. So you have to pay much attention about this summary of continuous and discontinuous variation. So the last but not least, let us also talk about the sources of variation. Crossing over, you have had the exchange of homologous chromosomes during prophase 1 of meiosis. This is very important because the effect of crossing over, it ends up introducing the new gene combinations within the population. Independent assortment at metaphase 1 of meiosis, you have had the movement of homologous chromosomes on the equator of the spindle fiber does not follow any pattern or any order. So that's why independent assortment at metaphase 1 of meiosis also brings discontinuous variation. Random fertilization of gametes. I have already explained this one. You know, during the time of sexual reproduction, gametes tend to fuse, the male gametes and female gametes fuse together. But now, how fertilization is done, it's a matter of a chance. It's a matter of a chance. The male gamete which is potentially capable is the one which fuses an egg cell. So, fertilization is done randomly. Again, there is random mating between individuals within a species. This is common in animals like dogs and pigs. Segregation of homologous chromosomes during anaphase 1 of meiosis. And again, there is what we call non-disjunction of homologous chromosomes. So when we say non-disjunction, here you have to understand non-disjunction is the failure where homologous chromosomes fail to separate during anaphase 1 of meiosis. So the end results here, it could be on the zygote which is going to be formed. The zygote could be formed when the zygote is missing some chromosomes, or the zygote could be formed when the zygote is having extra number of chromosomes. So that's how non-disjunction also brings in discontinuous variation.